Hi, welcome back to week two. This is chapter 4 on your schedule for June 9th's lecture. Here we're going to discuss cell membrane transport. So now that we've looked at the inside of the cell and its organelles, and we've completed chapter 3 on metabolism, our primary organelle was the mitochondria, we move back out to the plasma membrane and we're going to discuss the watery environment of cells. There's water on both sides of the cell membrane. So we're going to go back and review some things from chapter 2 where the plasma membrane was first introduced. And we're also going to see that large proteins inserted into this membrane add functionality to what would otherwise be like a droplet of oil floating around in water. So cells aren't just passive floating objects in a watery world. They are machines and they have function, and proteins are going to help determine functionality. So our topic outline here on this first slide, we're going to discuss the extracellular environment, or the ECF compartment, which is outside of the cell, also referred to as interstitial fluid, and then talk about movement across the plasma membrane. So things like osmosis, diffusion, facilitated diffusion, and active transport are all topics we're going to discuss here in this chapter 4 note set. Another thing that we will introduce here is a concept that we will use a lot in Unit 2. So your book, before it jumps into the simple idea of osmosis and diffusion, takes a little sidebar, and I want to make sure and walk you through that because these are concepts that are going to be really important in Unit 3 next week. So it's a good time to be reminded of the chemical nature of the cell's plasma membrane. We can see that here. It's made of a phospholipid bilayer. It has dual solubility, meaning the phosphate heads are going to be fully soluble in water and will point outwards and inwards to the two watery compartments in the ECF and the ICF. So ICF stands for intracellular fluid, ECF stands for extracellular fluid, and extracellular fluid can also be referred to as interstitial fluid. The long chain fatty acids, those tails point towards the inside. So you have this layer, this bilayer. Phospholipid bilayer makes up our plasma membrane. And it's going to act as a barrier and kind of be the outer border of our cell. The fatty acid tails are hydrophobic and they're water-fearing, so they don't want to be touching the ECF or the ICF compartment. This dual solubility feature is referred to as amphipathic. That's one way we can describe a phospholipid bilayer. And then you can see the large embedded proteins. They give the cell specificity when trying to transport substances into and out of the cell. Okay, so this is a look at table 4.1. And we'll be playing with the idea of molecules moving around in solution, and those solutions will represent the fluid compartments in the human body described in Chapter 1. Homework 2 has you drawing a human eukaryotic cell and identifying the fluid inside and outside of the cell, and also labeling the organelles. So these two fluid compartments are described here in Table 4.1. Notice they're distinct from each other. They're distinct because they're separated by a plasma membrane. So in this table, you have these lists of ionic values. So it becomes obvious that a healthy cell is not simply in equilibrium with its fluid surroundings. There's a difference maintained across the plasma membrane. So on the left, you have solute. What type of solute? Sodium, potassium, magnesium, calcium, on and on and on. There's a whole list. And then the ICF, or intracellular fluid compartment, in the middle column, you have 140 millimolar of potassium, 15 millimolar of sodium, 0.8 millimolar of magnesium, and on and on and on. Compared to the ECF, the column on the right. So if potassium has 140 millimolar inside, it only has 4 millimolar outside. So there's more potassium inside the cell than outside the cell. If we go down to sodium, where we have 15 millimolar inside the cell, we have 145 millimolar outside the cell. So the cell doesn't have a lot of sodium inside. And if you've ever had a really salty meal and maybe you're a little bit swollen the next day, you know that consuming sodium causes you to retain water. We'll talk about that again when we get to osmosis. But the cell has very, very low concentration of sodium inside or in the ICF compartment relative to the ECF compartment. So just taking these top two ions, sodium and potassium, as an example, we see huge differences between the concentration of potassium inside the cell and the concentration outside the cell. So how is this possible? We need to kind of think about that, have that question in the back of your mind as we move forward and talk about how a cell would maintain these different ion concentrations. Well, first we need to consider the cell membrane. This is a review from chapter 2. We were looking at cell structures, and the membrane's acting as a barrier, but it's not a solid brick wall. Substances can get through the plasma membrane. We've looked at a video for demonstrations on diffusion through a semi-permeable plasma membrane. This is a key feature demonstrated in lab number 2 that you're doing all week this week. So these concepts are important and definitely applicable. The plasma membrane is permeable to small, nonpolar molecules that don't carry a charge. So ions, although they're very small, are not permitted to simply diffuse across the plasma membrane. We will look at several methods of membrane transport here in this lecture. Some require ATP energy and are considered an active process, and others, like osmosis and simple diffusion, don't require any cellular energy and happen spontaneously, like rolling a ball down a hill. And that's one of the reasons why Table 4.1 listed out those ion concentrations for you. There's a natural tendency for things to want to flow from low to high on their way to, from high to low on their way to equilibrium. So an area of high concentration will flow to an area of low concentration. We'll look at that here in just a second. In this diagram, I've got some red arrows pointing to some components in your phospholipid bilayer, which is acting as a barrier for diffusion and osmosis. So we have the phospholipid layers that are in two layers, like a little sandwich. And we can see that there's some cholesterol inserted into this membrane to act as something to stiffen up the phospholipid bilayer. And then we see this large purple protein inserted into the membrane, adding functionality. And our main function, if we're a barrier and we're a plasma membrane, is to regulate what comes into and out of the cell. So here we are in figure 4.1. And there are many factors that govern the direction of molecular transport across the plasma membrane. So we've got some molecules in and out of the cell shown here in red spheres. Once we have a concentration difference across the membrane, so you can see in panel A, there's a higher concentration of these red molecules inside the cell relative to outside the cell. So the ICF compartment has a high concentration, let's call it 100, and the ECF compartment has a lower concentration of these red balls, and let's say it's 1. So you have a gradient set up from 100 inside to 1 outside. You could plug in whatever numbers you wanted to here. So there's going to be this chemical concentration driving force that will spontaneously drive substances from an area of high concentration towards an area of low concentration, regardless of whether it's into or out of the cell. If you look at panel B, the opposite's true. In the ECF compartment, we have a lot of red molecules trying to get into the cell where there's not very many. So spontaneous movement is going to tell us these two concentrations are going to want to go to equilibrium. That's a natural tendency for things to want to go from high to low until they hit their equilibrium point. So you can practice this idea by drawing a cell on your notebook paper and then plugging in the numbers given to you in table 0.1, which we just discussed. Once you've drawn your cell on your paper, plug in the numbers for sodium, potassium, magnesium, and calcium, and then draw an arrow, just like you see here in figure 4.1, towards the area of low concentration. This arrow indicates natural tendencies for diffusion. The arrow tells you which direction the movement of the molecules are going to go. Okay, so in passive transport or simple diffusion, I've got this movement of molecules down a concentration gradient from high to low, and this doesn't require any ATP energy from the cell. So things like diffusion, osmosis, even filtration, and facilitated diffusion, these are all passive. The cell doesn't have to put in any energy. The opposite is true when we look at active transport. This is going to occur during transport of molecules across the membrane using one of those proteins we saw in the first slide. So proteins add functionality, and some membrane proteins are able to use ATP energy to push substances in and out of the cell against the concentration gradient, and that requires work. So one of the things we can consider when looking at these small molecules is that sometimes these small molecules carry a charge. And if they're a charged particle, they're an ion. So charged particles are definitely going to be kept out of the cell. Ions will hit the plasma membrane, and it will be like a barrier. So the membrane's not going to allow charged particles to just simply diffuse across the cell membrane. That now allows us to consider the plasma membrane as a capacitor. Yeah, tiny human cells can store a charge. So what we see here in figure 2 is on the left, the ICF compartment. There's a plasma membrane barrier separating the compartment in blue. On the right, which is the ECF compartment. And we saw the species and its concentration value plugged in table 4.1. And anytime in the human body you have inorganic substances, they tend to go into a charged state. So calcium and magnesium and some of the common electrolytes you've heard of take on a charged state at pH 7, which is normal for human body pH. So they tend to be charged. And what happens is with the concentration differences across the plasma membrane, you also have separation of charge. So under normal resting conditions, the ICF, shown here on the left in figure 4.2, has a slightly negative charge, and a negatively charged particle is called an anion. In the ECF, we have an excess of cations. So charged positive particles on the outside of the cell, and that must mean the inside of the cell tends to take on a slight negative charge. And that's really because of the differences we saw when we looked at the list in 4.1 of the concentration differences between sodium and potassium. We'll talk about this in much more detail as we move on into Unit 2. We'll come back to this idea of separation of charge. So what this means is not only does the chemical concentration affect the movement of molecules from high to low, but the charge can create a driving force too. A difference in electrical potential will be measured in volts. It's established across the majority of human cells, and the ECF and ICF compartments are not electrically neutral. The separation of charge is measured as a cell's membrane potential, and we will actually measure this next week during lab number 3. So the separation of charge sets up an electrical driving force. And this is shown here in figure 4.3, where we can see the inside of the cell maintains a net negative charge, and those positive cations are kicked out of the cell, and under normal conditions, the outside of the cell is going to be positive. So the separation of charge can also drive movement because opposites attract. Okay, the charge difference across the membrane is also going to be another method of driving force across that membrane. Okay, so opposites attract. The positives are attracted to the interior negative, and the negative interior is attracted to the positive outside, and those charges tend to line up at the barrier. They can't get through, so you have this buildup of charge inside and outside of the cell. We will definitely talk about this a lot more in Unit 3. Unit 2. The next unit after Unit 1. Let's look at how molecules will be moving through these proteins that we inserted into the plasma membrane. So diffusion through protein channels, facilitated diffusion. Okay, this is still a passive process that uses a protein inserted into the plasma membrane, and it acts as a helper in order to facilitate the movement of molecules across the plasma membrane, which is acting as a barrier. So charged ions must have a protein channel assisting them across the plasma membrane, or they won't get through. This large protein is structurally similar to a barrel and will change form, and it will go in the closed position and the open position. So you can see here at the top, the form of the protein is sort of blocking or pinched in the middle, and the other formation just below it is in the open position. And whenever this protein's open, it's like punching a hole in the membrane, and molecules are going to simply flow from an area of high concentration to an area of low concentration, but they have to move through this protein, which is acting as a facilitator. It's helping the passive transport of these ions. This closed position is going to help regulate entry and exit in and out of the cell. Rates of diffusion are described for you by Fick's laws, and we will be looking at these in lab as well. So if you want to go ahead and take a look at the toolbox on page 103 titled Fick's laws, it describes the rate of diffusion and how it's dependent on a couple of important factors like permeability, solubility, concentration, how far does the molecule have to travel, so distance, which is going to be constant in human cells, surface area, probably also another constant in human cells, temperature, and molecular weight. If we look at carrier-mediated transport, it's a similar process, but here the membrane protein will bind with the molecule trying to get across, which will induce the protein to change shape temporarily and then move the molecule across to the other fluid compartment. Polar molecules or very large molecules cannot simply diffuse across the plasma membrane. They are transported across by protein carriers. This sets up a point of regulation. Fick's laws says the rate of diffusion depends on permeability, concentration, distance, surface area, temperature, size and solubility of the molecules trying to pass. Anytime you have a carrier-mediated transport process, you must determine if there is any ATP energy requirements. Facilitated diffusion is a passive process, but active transport is an energy-requiring process and is ATP-driven. Some protein carriers will be able to burn an ATP in order to have the energy to push molecules around in an active process. Protein carriers exhibit specificity for a single molecule, usually due to van der Waals and hydrogen-hydrogen bonding. Competition in the available substrates for transport will occur at the protein site, and protein carriers often exhibit saturation, which is when all the carriers are occupied and therefore reach a transport maximum, and that's shown here on this graph, where you have a logarithmic phase and then it tops out and it flattens out at the top. The protein carrier simply can't transport anymore. This is figure 411 in your textbook. Okay, so facilitated diffusion. If we look here in figure 10, we get to see how the protein interacts with the substrate. In this case, it's a glucose molecule. This is where large, bulky molecules, like our 6-carbon sugar, are going to be bound by the protein temporarily, causing the protein to change shape, and when it changes shape, it dumps the sugar molecule to the inside or ICF compartment. of the cell. So glucose comes from outside of the cell, binds with its protein carrier, causing a change in conformation, and now the protein's open to the inside of the cell. The intracellular fluid is this tan compartment down here at the bottom of these pictures, and you can see that the glucose molecule is going to float off into the inside of the cell. So this is some specificity involved when we talk about moving sugars directly into the cell. When we get to Unit 3, we'll look at how insulin can play a part in glucose transport. Now, if we switch gears to an active process, active transport systems include primary and secondary active transport. This is the transport of a molecule against its concentration gradient, and this is going to require ATP as an energy source to do this kind of uphill work. So the transport movement in this form is from low concentration on the left, movement towards an area of high concentration on the right. So we see many more red solute molecules on the right, or in the ECF compartment, than we do on the left inside the cell. From moving from low to high is an uphill push, and it requires ATP energy. This protein has to be able to cleave a terminal phosphate off of ATP. When it becomes phosphorylated and ATP donates a phosphate group, the protein's now energized to do this kind of uphill work. Another common example of primary active transport is the sodium-potassium pump. It's a pump because it's going to push sodium and potassium against their concentration gradient in both directions. This protein actively kicks out or extrudes 3 sodiums out of the cell against the concentration gradient. When it's in the open position towards the extracellular fluid, after releasing sodium, it will bind potassium 2 at a time and bring them into the cell against their concentration gradient. You can prove these numbers to yourself by referring back to Table 4.1, where the millimolar concentrations for sodium and potassium were given. This protein hydrolyzes an ATP for energy, one directional movement of both ions. So when it goes back to the resting position, open towards the inside of the cell, in the lower left panel, you can see potassium drifting off into the cell as it's being pushed uphill into its area of high concentration. This is going to set up a steep concentration gradient. Again, listed for you in Table 4.1. Those differences in concentration across the cell membrane is referred to as a gradient. So you can have coupled transport, you can have electrical impulses. We're going to be looking at action potentials in nerve cells here in the next unit. And we can also help regulate cell volume with this type of movement. Water likes to follow salt. That's why salty meals and high sodium diets often helps us retain a lot of water. So we'll look at that with osmosis here in just a second. Let's look at secondary active transport. So remember, anytime you see the word active transport, ATP is in the word. So here we have this coupled transport. It requires ATP, so it's an active process, and it moves more than one substance at a time. It couples endergonic with exergonic activities here in Figure 4.15. We can have co-transport, which is referred to as a symport. Both molecules are moving in the same direction. Here in panel A, glucose and sodium are both being moved together. The downhill tendency for sodium to want to be inside the cell provides that slingshot of energy to bring a glucose along with it. Again, the protein would have to be able to bind and associate with both molecules in order for this to happen. But the downhill energy of sodium going into the cell is going to give the energy to bring along a glucose. So this is a symport or co-transport. The panel next door, panel B, is a counter-transport or antiport. That is where molecules are moving in the opposite direction. So glucose transporter is another example of an energy requiring movement coupled to the downhill movement of sodium into the cell. This is going to help drive the flow of glucose into the cell, and the sodium glucose transport protein has an affinity for both molecules and will bind them specifically. This is shown again for you here in this picture from your book, where you can see the proteins binding both molecules and transporting them specifically. Let's switch gears and talk about osmosis. So the specific movement of water. This is a passive movement, doesn't require any ATP energy. And the movement of water across the cell membrane is important to consider in human physiology. The flow of water into a cell, when in this case a red blood cell placed in pure water, can take on large amounts of water, and this filling of the ICF compartment can continue until the cell ruptures, causing cell death. So in figure 417, we see this tendency of water to want to rush into the cell, that large arrow pointed in towards the solutes, to equilibrate water and salt. So that's why I said earlier, water tends to follow salt. So when you put a cell inside of pure water, there's no electrolyte or ion concentration in pure water. You have 100% water and maybe 90% water inside the cell, because there are going to be dissolved solutes and proteins inside the cell. Water is definitely going to want to rush in. The opposite is true if the cell loses too much water and then shrinks to the point of cell death. In the lab, we would use an isotonic solution to prevent cell damage from osmosis. Hypotonic solutions are low salt content, high water relative to the ICF compartment of the cell. So the cell reacts to these conditions and starts to swell as water rushes into the cell. The opposite is going to happen when a cell is placed in a hypertonic solution. This is a solution with high salt and lower water content than the cell. The cell will lose water to its surroundings in this situation. Figures 417, 418, and 419 will help you with this concept, and there will be several videos that will put these molecules into motion. So the concept of tonicity is the idea of the cell having an effect when it's put in different types of solutions. So tonicity is the solution's ability to affect change on the cell. So defined for you here is isotonic, hypertonic, and hypotonic. You have these in your notes. And I'm also going to point you in the direction of page 113. There's a toolbox specifically on osmotic pressure, and that toolbox will help you get through the slides that follow this one, that I'm going to let you guys study these on your own to show that there is power in water. If we look at compartmental transport with this idea we've been painting for movement across the membrane, we've talked about small molecules, but what about larger molecules? Those are going to be moved through vesicular structures across the membrane. So if you remember what the micelle looked like in chapter 2, we come back to this idea where we see sort of a pinching off of the plasma membrane so we can transport big molecules. So endocytosis and exocytosis will be discussed here. This is figure 421, and it's a method of moving large, bulky molecules into and out of the cell. Cells can incorporate large macromolecules with the assistance of membrane proteins and micelles, which act as transport vesicles inside the cell. Phagocytosis is when large phagosome engulfs ECF particles and helps bring them into the cell for processing. Pinocytosis is a spontaneous pinching off of the membrane, creating an endosome that contains ECF fluids and small particles from the ECF compartment or interstitial fluid compartment. Down at the bottom in panel C is receptor-mediated endocytosis. This is a highly specific process where integral membrane proteins called clathrin act as receptors that recognize and bind certain particles in the ECF compartment and bring them into the cell, which also helps determine the cell's response. Exocytosis is the endocytosis process in reverse. So here we can see in figure 4.2.2, the cell is kicking out unwanted large particles. So exocytosis allows the cell to expel large bulky molecules that are no longer needed by the cell, or molecules destined to do their work outside of the cell. So endocytosis in reverse. Okay, I'm going to stop here with your lecture notes for chapter 4. Make sure you utilize the videos on this page from other resources that I've pulled together for you. And there are also videos on diffusion in your lab number 2 assignment. Okay, thanks for joining.